Research is the cornerstone of innovation in clinical practice, driving improvements in patient outcomes and shaping the future of medicine. Welcome to the Global Rhinology Network Topical Research Symposium. I'm Ashok Rokde from Winchester, UK, and it is my privilege to welcome you as a host. Joining me as co-hosts are Louise Melia from Bath, UK, and Professor Thibaut Van Zelle from Ghent, Belgium. This symposium showcases fascinating topics across 4 key modules: General Rhinology, COVID session, and Research in Chronic Rhinosinusitis. A special highlight is the PhD session featuring research conducted by enthusiastic young rhinologists who represent the future of our field. This symposium brings together leading global minds in rhinology who will highlight latest developments with a focus on translating research into impactful patient care. The Global Rhinology Network is dedicated to advancing knowledge innovation. Thank you for being part of this exciting journey. Thanks to Medtronic and Karl Storz for their generous support in making this symposium possible. Welcome to GRN-Topical Research Symposium.
Our penultimate speaker tonight is Ines Lammens, who's currently a PhD fellow and ENT resident in Ghent University, having started a PhD in October 2020. And tonight is going to be telling us about their project on elucidating mucosal IgA responses in allergic airway disease.
Thank you very much for the kind introduction. Can you see my screen?
Yes. Perfect.
I would like to thank the organizers for giving me the opportunity to present my PhD here. So I'm a MD, ENT training at the Ghent University Hospital, and I'm currently doing a PhD in the lab of Professor Philippe Gevaert and Professor Bart Lambrecht, and I'm looking into mucosal IgA responses in allergic airway disease. Now, the airway or the respiratory tract is actually daily exposed to millions of particles via inhalation. And the airway epithelium itself serves as a physical first barrier against possible airborne pathogens. However, in the context of allergic airway disease, we currently do not know how protection of this mucosal layer is governed. Now, another site in the body, a mucosal site that is also chronically exposed to antigens, is the gut, so the intestinal epithelium actually. And here, protection of the mucosal layer is governed via Peyer's patches. These are local germinal centers or local lymph nodes actually within the intestinal epithelium, where IgA plasma cells are being produced through a germinal center reaction. Here, germinal center B cells proliferate into IgA-producing plasma cells, and then this IgA is transduced over the gut epithelium, and there, secretory IgA maintains several protective functions to protect this intestinal barrier, epithelial barrier. Now, interestingly, upon viral infections, this is a mouse study actually that was recently published. And in this study, it is shown that following viral infection with an influenza strain, IgA levels in the bowel fluid of mice, so in the bronchoalveolar lavage of mice, is increased. Now, interestingly, in allergic patients, so allergic rhinitis and allergic asthma patients, local IgA is also increased compared to healthy controls. And so this suggests or shows that IgA antibodies are actually enriched in the airway. Now, look in the gut, these antibodies, as I mentioned, are being produced by a local germinal center reaction just below the epithelium. Now, in the airways, tertiary lymphoid organs, or TLO, are induced upon chronic antigen exposure. And these TLO, they resemble SLO, or lymph nodes thus, in organization. They consist of a B cell follicle with a germinal center and an adjacent T cell zone. In this germinal center, this could give rise to the formation of plasma cells and memory B cells, which could then perform protective functions within the tissue, so within the airway epithelium. Now, we wanted to understand these mucosal responses actually, and whether IgA is being produced through these TLO, and therefore we challenged mice. So we exposed them chronically to house dust mite. House dust mite is the major allergen to which allergic patients are allergic. And we exposed these mice chronically to house dust mite. We sensitized them with a low dose house dust mite. And then we come back with higher doses of house dust mite on a chronic basis. So 3 times a week during 5 weeks. And when we look into the lungs of these mice, they show evidence of TLO that are being induced. And this we can show by the phenotype of germinal center B cells locally in the lungs. Prior to sacrifice, these mice were injected with CD45 antibody, and this is to discriminate blood from lung tissue cells, actually. And so here we see a nice germinal center B cell population, which is a proof of a TLO that is being induced in the lungs. On the kinetics here, you nicely see that these TLO are induced starting from 3 weeks of house dust mite challenges, and with additional challenges, they increase further. In orange, the MLN, or the draining lymph node from the lungs, is shown. This is a positive control. So this is the mediastinal lymph node as a positive control. Now on imaging, we can nicely see these TLO actually. So this is the airway of a mouse that was challenged for 5 weeks. And here just below the airway epithelium, also over here and this area, you see these accumulations of cells just below the airway epithelium. When we zoom in into this site, we see that this accumulation is actually a B cell zone because it stands for B220, which is a marker for B cells. And in this B cell zone, you have GL7 positive cells, which is the germinal center, actually. In this germinal center, you can also find follicular dendritic cells. These cells present antigen to the B cells in the germinal center. And dispersed in the B cell follicle, you see T cells actually. Now, interestingly, when these mice are challenged chronically, and so they induce TLO in the lungs, we also see an enrichment of mucosal IgA in the bowel fluid. So this is done by ELISA, and you nicely see that IgA is actually enriched in the airways. This is for polyreactive, so the total IgA, but also house dust mite specific IgA that increases in the bowel fluid. And interestingly, over time, inflammation decreases in these mice. So eosinophilia locally in the lungs is a sign of Th2 inflammation, which is an allergic inflammation. And after 1 week, these mice are allergic. They have around 2 million eosinophils. This increases further, and then from 3 weeks on, the eosinophils locally, they drop. This kinetics is also shown by the IL-4 kinetics locally in the bowel, IL-4, which is a prototypic cytokine for Th2 inflammation. And so interestingly, these mice, they induce TLO, they are enriched, the lung mucosa is enriched for IgA, but inflammation is down. And so we wondered whether these TLO are actually implicated in this local IgA production. And so looking into the germinal center B cells further, when you stain for IgA and IgG1, you actually nicely see that within the lungs, there are IgA positive germinal center B cells, which are not present in the lymph nodes. Again, looking at the kinetics, you see that IgA class switching recombination, which is actually a B cell that goes from IgD to IgA to become a plasma cell or a memory B cell, is only taking place in this lung germinal center. We can also stain for that on microscopy. So this is the consecutive section from the same mouse. Again, you here have the TLO, and in orange, in pink this time is IgA that is being shown. And as you see, just below the epithelial, IgA positive cells are actually enriched, which suggests that IgA positive cells are just lying under the airway epithelium. Again, if we zoom in, we can see here that within the germinal center, there is indeed these are signs of IgA class switching in the lung TLO. Here you can see these halos of IgA. These are actually IgA producing plasma cells that are lying just beneath the airway epithelium of these mice. When we can also prevent that these TLO are being formed by injecting the mice with lymphotoxin beta receptor antagonists. And this, as you can see here, so these mice are chronically treated with house dust mite. And when they are injected with these antagonists, their population of lung germinal center B cells, thus the TLO, is decreased. Interestingly, this goes along with a decrease in mucosal IgA of the lungs of these mice, both for polyreactive as for house dust mite specific IgA. And most interestingly, inflammation is again increased in these mice. So this suggests that when TLO formation is prevented, the mucosa is no longer enriched for IgA and inflammation increases again. To conclude my work at the moment, we have shown that chronic antigen exposure with house dust mite induces TLO in the airways of mice. This presence is accompanied by an enrichment of mucosal IgA in the airways itself. This IgA production is mainly taking place in the local germinal center of the lungs, so in the TLO. And when this TLO formation is prevented by injecting mice with lymphotoxin beta receptor antagonists, mucosal IgA is decreased and inflammation increases again. So to end, I would like to thank everyone involved in this research, especially Professor Bart Lambrecht and Hamida Ahmad Stijn, who's my co-promoter and is really, true guiding person. Then Josephine, who just presented, she's also involved in this research. Antonio, who taught me his way of doing microscopy. And of course, the people from the Upper Airway Research Laboratory at UGent, Professor Philippe Gevaert, Nathalie and Gabrielle for doing laboratory work. Thank you very much. I'm happy to take questions.
Thank you very much, Ines. That was excellent. Does anybody have any questions at all?
Can I ask a question? The fact that your TLOs are located just next to the epithelium, do you have an explanation for that?
Well, as I've shown in my research, in the presentation, these TLO are an organization of B cells and T cells, and they seem to produce IgA plasma cells. So it would make sense in line with what is from the gut Peyer's patches, which are also just below the epithelium, that they are close to it, because then IgA can easily actually enter the mucosal space.
Yeah, but to develop the TLOs, is there something that might be secreted by the TLOs that is inducing TLO formation?
That's actually a very interesting question. We are currently looking into that. What drives this accumulation and especially this organization of B and T cells. You can also in the lungs, but I didn't show, you have also B cells that accumulate around the blood vessels in the lungs, but there you have less germinal center activity. So there's something about where they are located next to the airways that is favoring or promoting this organization actually. But we are also curious and we are looking into that.
Okay.